Good evening, everyone. I'm Sebastian Lay, and this is probably the earliest we've gotten started this summer. So we're not going to. I had a freshman professor who would lock the doors right at, on the hour before you could get into the lecture hall. And so I think we can never be that strict, but thank you all for coming. So I think we're in for a real treat tonight that spans actually the whole village, if you will. So it's a particular pleasure this late in the season as the currents are warming up around here, and I think more and more of us are spending more and more time on and in the water. So on that note, take these out and turn them off. Otherwise, the water is nearby. But it helps us to enjoy the lecture, so please do it. Additionally, tonight's lecture is both recorded and simulcast, just for your information. And after the lecture, there is a Q&A. If you don't have time to ask your question, please join us in the Meg's room in the Swope Center, which is upstairs in Swope. The crowd will lead you, and we can continue the conversation there. Finally, as this is the penultimate lecture of the season, I start to get a little nostalgic and say a few things about the tradition that these lectures represent. Over 130 years now of intellectual exchange in Woods Hole, previously in a much smaller space, I assume, now in this storied auditorium, and you all are a part of that. So thank you. Wind down your summers. And with that, I will once again remind you of your cell phones, but also first thank and introduce Jack Costello, who is a longtime MBL investigator, but importantly is currently chairing our Whitman Scientists Steering Committee and volunteers a lot of his time in guiding the summer scientists community here, advocating for their work, and really advocating for the MBL. So please welcome Jack and our speaker this evening. Thank you.
Hi, everyone. It's a great pleasure tonight to have John Dabiri here. I want to give you a little background about John to place this talk. John is an expert in fluid dynamics, and fluid dynamics is very important to us as biologists because the animals that we study live in a fluid, and the study of fluid dynamics is the study of how those fluid interactions occur and how they influence organisms and how organisms influence them. And so we're very interested in it. One of the issues with dynamics is that it's often described in very mathematical terms. Differential equations and equations that kind of run on in a paragraph. And so for many biologists, they're more difficult to work with. Well, this is where John comes in as really a great bridge, because not only does he excel in the science of fluid dynamics, but he also understands and is interested in real animals. And so we've been lucky at the MBL to have him working with us during the summer for the last 14 years. So John's not a stranger to the MBL, and we're very glad to have him here. And this ability to bridge the range between advanced theoretical work and real with animal work has enabled John to really make very substantial and brilliant discoveries in the field, as you'll see tonight. And so that's what's led to that glittering resume that you may have read in the announcement for this, and is what also makes us fortunate to have him here tonight. So I'd really like to welcome John, and thank you.
Thank you very much. And thank you all for coming. It's really a pleasure to be here and to be able to share with you a work that was inspired by one of my heroes in science, Walter Munk. The other hero in science is Jack Costello, for that very kind introduction. Let's see if we can get this to advance here. So some of you will know Walter as one of the founders of modern oceanography. When I began my assistant professorship at Caltech, I actually hadn't heard of him, coming from a theoretical fluid mechanics background. But when I started my position, I quickly learned that the biggest challenge in being an independent investigator was not coming up with solutions to various research questions. It was trying to figure out what questions to ask in the first place. What would be a worthy topic to dedicate my career towards? And so for inspiration, I would spend time in the library, roaming the stacks, looking at some of the old journals to find some of the classic papers. For the youngsters, this is back in the olden days when libraries had books with paper and whatnot. So I came across a paper of Walter's. It's called Abyssal Recipes. So it's his ingredients for how the ocean works, how it functions, how it circulates. He talks in it about the winds and the tides and the effect that they have on ocean circulation. But in there, he also has this excerpt, which I've reproduced here. He notes the most abundant organisms in the abyssal sea, in the deep sea: copepods, mysids, euphausiids, krill, squid in the nectonic bristlemouth, small fish, are all engaged in daily vertical migrations, diurnal migration, over vertical distances of order 1 kilometer. And then I've highlighted this comment he makes, that this migration is associated with some transport of water. So for me, having an interest in fluid dynamics and biology, this seemed like a really exciting topic to think about. And I'll highlight it with this question: do swimming animals mix the ocean? An unlikely question maybe, but one that hopefully by the end of this talk won't seem quite so ridiculous. Now, before we begin, I should better quantify what I mean by mix the ocean. So we'll talk about it in two contexts. The first is, do these animals somehow contribute significantly to how the ocean circulation is maintained? This is, of course, important for global climate here on Earth. We know that generally speaking, water tends to cool and sink at the poles, and so some energy sources are needed to mix that denser water back up. If you lift something up, you need to provide energy to change its potential energy. And so we ask whether these animals might play a role in that process. Secondly, we ask whether the animals might significantly contribute to the transport of carbon, nutrients, oxygen in the ocean. Here I'm showing a classic sketch of what's sometimes called the biological pump, but the basic idea is that as we emit atmospheric carbon, for example, it gets sequestered in the deep ocean through biological processes, the food web, for example, but also through downwelling and upwelling in the ocean. And so the question is, in addition to winds and tides, could the animals play a role in these critical, important processes for global climate? Now, what do we know today? If you ask a card-carrying physical oceanographer like Ray Schmidt here in the front row, he'll tell you that there are at least a couple of important mechanisms. The first is the wind. It'll blow at the ocean's surface globally. That'll tend to create what are called internal waves. Think of them as basically like the surface waves you can observe at the sea surface, but at various depths between regions with different physical properties, different densities, for example. Now, earlier we heard that fluid dynamicists tend to go on and on with equations, so just a couple of numbers to remember today and you're off the hook. This order of about 1 trillion watts of kinetic energy per unit time, this is the kinetic energy flux that people will associate with that wind forcing, 1 trillion watts of power. And also, another way that we sometimes measure how much energy is being expended in this process is in a per unit mass framework. So how many watts per unit kilogram, per unit mass per kilogram of water. So 10 to the minus 5. So the only number you need to remember tonight is this 10 to the minus 5. I'll come back to that later. So one mechanism is wind. The other is the effect of the moon. Tidal currents over different features on the seafloor can also generate these internal waves which break and mix and can help to redistribute those ocean properties. Again, estimates here have pretty big error bars, but something a little less than maybe 1 trillion watts of power being put into the ocean by these processes. So what we also know from some back of the envelope estimates is that we need around 2 trillion watts of power to maintain global ocean circulation. And so it's conceivable that these two are sufficient. If you take this number, and you squint a little bit, it's about 2 trillion watts together. And so maybe winds and tides are the full story and we can all go home, although you guys just got here, so we'll continue. And think about animal swimming. Could that play a role in this process? Now, first of all, what do we know about where animals are located? This is actually what I would suggest is an outdated picture, but the most common one today, which is that we know light, of course, is greatest at the surface and decreases with depth. We expect that because of the food web, everything depends on photosynthesis. And so we expect animals to be biased toward the surface. And then later on, it's been discovered that there are also animals that inhabit the abyssal plains at depth. One of the important caveats about this picture is that it's very much biased by how we can collect our measurements. Our surface ships are all sitting up here, and so we have many more measurements of animals up here than we do at, say, 2,000 meters. The very few occasions where we do have the opportunity to, for example, take a remotely operated vehicle and go into deep water, we find that in fact there are a whole trying to do a census of the ocean, and I'm putting down a net and pulling up whatever comes out. These are goop by the time they get to the surface, and they're not going to end up in our counts. So the take-home message here is that, in general, we expect that a lot of the ocean can be populated with organisms. The number that comes to play here, again, is this 1 trillion watts. So Bill Dewar, a scientist at Florida State, made an estimate based on the total amount of chemical energy that goes into the ocean, about 60 trillion watts of chemical energy per unit time, how much of that goes into those swimming motions that they create. And you get about 1 trillion watts. So it's the same amount of power, roughly, as what goes into the winds and as what goes into the tides. So that was the first key that this might be a process that's important, biogenic mixing, as we call it, this biologically generated mixing. A second was a group, Huntley and Zhu, who basically did a theoretical study. They said, let's take everything from tiny krill to large sperm whales, and let's try to make estimates of how much energy they're creating or creating as they swim. And so you don't have to read this eye chart of a table. I just want to point out that if you take the mass of the animals, again, across a huge range, their abundance, so how many of these animals do we see per cubic meter in the water? How fast they swim, and then also the efficiency of their swimming. How much kinetic energy do they create in the water as they swim? You can come up with estimates of that dissipation rate. Remember earlier I asked you to keep a number in mind? What was that number? Awesome, everyone's awake so far. 10 to the minus 5 watts per kilogram. And you get the same from these theoretical estimates, that the kinetic energy dissipation rate, how much energy is being put into the water, is actually comparable to the wind forcing and the tidal forcing. Now, the paper that I would say drew a lot of interest in this field were the first real measurements in the field. So the ideas previously were largely theoretical or based on estimates from, say, net primary production. Here what we have are measurements. So this is 7:30 in the evening in Saanich Inlet in British Columbia. And at sunset, you're seeing a rising zooplankton layer. So it's rising from the bottom here at 100 meters to maybe 50 meters. As this migration of krill rose, they were able to directly measure how much turbulence was being created as these animals rose up. And what they found here, these red spikes, basically just indicate places where their measurements saw very large rates, even larger than before. And so for those who haven't been in math for a while, 10 to the minus 4 is actually 10 times bigger than 10 to the minus 5. So a large increase in the kinetic energy dissipation rates versus those other oceanographic processes. To be working on. And we were a couple of years into the research when the tide started to shift a little bit. That's a bad pun, but I'll use it. Kunze et al., the same group that took those earlier measurements, weren't able to subsequently find similar measurements at that location. And so they published a follow-up paper. This one was published in Science. When you disprove yourself, that doesn't get published in Science. But nonetheless, based on the sampling to date, turbulence production by the marine biosphere is not efficient. So, okay, maybe there's a mixed result there. Next, Andre Visser noted an important concept that we'll come back to in this talk. So far we've talked about how much energy these animals are putting into the water and expecting that, well, that's a big number. That must mean there's a lot of mixing happening in the ocean. But the important thing here is that this kinetic energy dissipation is not the same as mixing. If you take your cup of coffee in the morning and you pour in milk, imagine you took a normal sized spoon. It only takes a little bit of effort for you to mix the coffee with the cream. Imagine I shrunk down the spoon by a factor of 100, so it's this little millimeter-sized spoon. That would be weird, but if you did that, it really wouldn't matter how much energy you put into the coffee. You're not going to be able to mix the two together. So there's a length scale effect here that matters in how effectively you're able to mix. And so what, in fact, you want to focus on, as Andre points out, is what's called the mixing efficiency. How much of that total kinetic energy I'm putting into the water is actually going to affect the water column. And I won't go into the details, but you can come up with some relatively straightforward theoretical models of that efficiency as a function of some length scales that I'll come to in a moment. So first, on this vertical axis, this is the efficiency of mixing. Here at 10% efficient, that means that, let's say I put in 10 watts of power per unit time, 10 watts of power into the fluid. Only one of those watts is actually going to do any mixing. The rest is just churning the local water, eventually being dissipated as heat. And then on this log scale here, this is 1% efficient, 0.1% efficient, etc. Now, this curve shows us that the mixing efficiency increases as this ratio of a length scale to this buoyancy length scale changes. The buoyancy length scale is just a characteristic of the ocean. Think of that as a set number, a meter, something like that for our purposes. The L here is the size of the stirring eddies that these animals are creating. So going back to our coffee cup, that would be the size of that spoon that you're using to mix. So the bigger the spoon, the more efficient you are in terms of mixing. So what Andre shows is that, well, if you take the length scale and you assume these animals are creating little mixing eddies as big as their bodies, then the mixing efficiencies that correspond to them, their ratios are here between 0.1 and 0.001, are going to be at best 0.1%. So sure, Bill Dewar at Florida State said lots of energy is being put into the ocean. Sure, the dissipation rates might be the same as those other processes, but the length scales are too tiny to matter. Basically, if the length scale is limited to their body size, the viscous effects in the ocean mean that they're not really doing anything for us. So at this point, I would say the verdict was sort of split. You have some anecdotal evidence saying maybe animals are important. You have other evidence that says they aren't. You have some theoretical models that say, yes, this is important. You have other models that say they aren't. In the field, though, people were generally very resistant to this idea. I, on the other hand, was all in. And, of course, I was not biased. I had spent 2 years thinking about the problem. But I went back to that interaction that I had in the library, reading that paper by Walter Munk. And so I said, you know what, I figured it out. I'm going to email Walter. And he's, one of the gods of the field. He and I will collaborate. We'll set the field straight. I was imagining the papers we'd publish together, Dabiri and Munk, or Munk and Dabiri. I'd let him pick which he preferred. And it would be great. I'd get tenure, happily ever after. So I emailed him, and he, in fact, replied within the hours. And I thought, of course, he's probably as excited about this as I am. He's pumped to get going. You can see here, this is the email I sent to him. I was a little bit tentative. So my subject line was ocean mixing by animals, ? But nonetheless, I asked him what he thought, and I was excited until I started reading. So he says, "Dear John, it was partly an attempt at humor when I suggested many years ago that diurnal migration could lead to appreciable mixing. And I was amazed at recent papers who think this is not a joke." So I remember sitting there in my office and staring out the window and thinking, so this is how my career ends, I guess. I'll go back to Ohio. My mom doesn't know what I do anyways, so she'll be happy to have me home. But fortunately, his email didn't end here. If it just said, "Sincerely, Walter," that would be a terrible email. He continued, and he says, "And people thought it was a lunatic idea," or in Carl Wunsch is an oceanographer at MIT, suggested that the moon, via lunar tides, remember that was one of those two components that is well accepted today, that could have anything to do with mixing, and now that's generally accepted. I must say we prefer the era when we were called lunatic to the era where people say, we all know what's happening. And then he ended with advice that has really shaped my career. He says, I can only say work on problems you most enjoy. Strange things can happen underway. And so with that advice, we forged on, and we had two main objectives. The first was to try to get direct measurements of this mixing efficiency in the water, to not try to rely on these theoretical models or even the shipboard estimates where you're not directly seeing what the animals are doing, you're just getting those turbulent fluctuation measurements. And then again, finally, to figure out what are the physics at work in this process. So as Jack mentioned earlier, in collaboration with him and Sean Colin, we've had a great time studying various jellyfish systems, and primarily my initial motivation was in understanding propulsion. So we work with the Navy, for example, trying to design more efficient underwater vehicles by taking advantage of some of the mechanisms that these organisms use. But as you can see, in a place like Jellyfish Lake here, they come in huge numbers, and so this seemed like an ideal context in which to think about how they might be able to mix the local water. Now, to study this, I was fortunate to come across a fantastic student, Kakani Katija. She's now a principal engineer at the Monterey Bay Aquarium Research Institute. Kakani grew up wanting to be an astronaut, it turns out, and so she went to the University of Washington as an undergraduate to study aerospace engineering. Along the way, turns out she's a very good figure skater, so she was on the U.S. national figure skating team on the side. Slightly more impressive than my own hobby of fantasy football. Although I won the league last year, I will say. It's a true story. So she's a principal engineer at MBARI, but what she did for her Ph.D. with me at Caltech was to think about how we could develop technologies that would allow us to measure the flow around these animals, as Jack mentioned earlier, to do it in a way that was quantitative. The water itself, of course, is transparent, so we'd like to come up with ways to be able to quantify how the animals are interacting with the surrounding water. So the work that Jack and Sean pioneered is the use of these dye-based visualizations here. And so I use this to show the kind of flow around these animals. Think of it at your local aquaria. They usually don't have the ability to show you this kind of flow. It makes these jellyfish maybe the most exciting organisms in the aquarium, in my opinion. Look at the flow that they create when this dye is released here, these swirling vortex rings in the wake. As they swim. These are their feeding appendages here. And so what Kakani did in her thesis was to develop these technologies that would allow us to make even more quantitative measurements of what the local flow is doing. So this is her in the Caltech swimming pool early on, Jack and Sean in the water. You may be saying, John, why aren't you in any of these photos? We'll come back to that. What we'll have Kakani do is tell you a little bit about the design of the system so you can get a sense of how this works. This is her at a TED Talk.
And so a single scuba diver can go anywhere from the surface to 40 meters or 120 feet deep to measure the hydrodynamic signatures that organisms create. And now I want to, before I begin, I want to kind of immerse you into what these kinds of measurements require. So in order to work, we actually dive at night, and this is because we're trying to minimize any interactions between the laser and sunlight. And we're diving in complete darkness because we do not want to scare away the organisms we're trying to study. And then once we find the organisms we are interested in, we turn on a green laser. And this green laser is actually illuminating a sheet of fluid, and in that fluid, it's reflecting off of particles that are found everywhere in the ocean. And so as an animal swims through this laser sheet, you can see these particles are moving over time. What you're going to see is that on the left, these are particle images that shows the displacement of fluid over time. And using that data, you can actually extract what the velocity of that fluid is. And that's indicated by the vector plots that you see in the middle. And then we can use that data to answer a variety of different questions, not only to understand the rotational sense of that fluid, which you see on the right, but also estimate something about energetics, or the kinds of forces that act on these organisms or on the fluid, and also evaluate swimming and feeding performance.
So with this tool in hand, we now could take very precise measurements. This is in Long Beach, California. The animal's glowing green because of the laser. The animal's not being hurt, I promise, in the process. And what we can do then is each of these arrows is telling you locally how fast the flow is moving and in what direction. So combining these measurements with some additional measurements to study how the density around the animals, the fluid density is changing, we were able to quantify the mixing efficiency directly. And the short answer here, I won't go into the details in Q&A, I'd be happy to, the animals were showing efficiencies on the order of 20%. 20 to 30%, as opposed to that theoretical model by Andre Visser, which would have suggested 0.2%. 100 times higher efficiency than you would have expected from that model. And so at first I thought it was wrong and said, Kakani, you got to go back in the water. And she said, why don't you go in the water? We'll come back to that. But this turned out to hold and to be a robust result that later was also supported through numerical simulations. So the question was, okay, well then what's going on physically that's leading to this very high efficiency of transport in the ocean? So one way to think about this is to go back to these videos that I showed you earlier. And the prevailing notion was that the animal, it creates those swirling vortex rings, those smoke rings like that you would see behind a cigar, for example, and that those are mixing the water. The challenge, again, is that the length scale of those vortex rings is roughly the same as the length scale of the animal. You put that in Andre Visser's model, you get a low efficiency. So there's some inconsistency there. If you stare at these videos long enough, you'll see something else happening, though. Train your eye behind the animal's oral appendages here, and you can see that this dye that we've injected keeps getting pulled along with the animal as it swims. I'll show this video one more, and then we'll see a different perspective. So it's pulling along that fluid. This is a better perspective because now we're going to be moving with the animal, and you're going to see as the dye is injected, it's drawing out this filament of dye here, pulling it along with it as it swims through the water. One more way to see this, using that technique Kakani described, these are just little microspheres that we put in the water column. If you look by eye here, you can see how the water behind the animal, with these suspended particulates is being pulled almost like a rigid mass behind the animal, even as the animal, of course, has to propel water backwards in order to swim forward. So this induced drift effect, we thought, could turn out to be the key to why these animals are so much more efficient than you would expect. And we called it a Darwinian mechanism because it actually comes from this family. Now, of course, when I say Darwin, especially at a place like MBL, everyone wants to jump to the author of On the Origin of Species. In fact, just a fun side note, the title is actually much more ambitious. It's On the Origin of Species by Means of Natural Selection or the Preservation of Favored Races in the Struggle for Life. So a very august title. His grandson was a physicist who decided to go with the more modest title Note on Hydrodynamics for his famous work. So in this paper, which turns out to be his most famous work and the piece of information that was really critical for our research, he studies this question of what happens if I have an object that moves through a flow. Potential flow, don't worry about the jargon here or the equations, this was just for Jack. It's the idea that something's moving through water and we have a simplified fluid model. We're not gonna worry about viscosity or some of the other physics of the system, just the pressure that it exerts. And what he shows is that when you have an object move through fluid, and imagine these are all water molecules we're tracking or water parcels we're tracking, that there's an initial forward motion, the particles tend to loop to the side out of the way, and then there's this permanent forward displacement. So things get pulled along with an object, even without any viscosity in the water. Now, lest you think this is just a cute theoretical construct, I found this on YouTube. This guy's vaping, and he releases smoke near the semi, and you get the same type of looping trajectory occurring there. Now, I have to add the caveat here. I don't recommend vaping or standing by the side of a semi, or you might demonstrate that other Darwinian principle of natural selection. But it is interesting to see that this is a real physical effect that occurs. So the object moves. It creates this drift volume here. And the interesting thing from Darwin's work is he shows that the ratio of that fluid that's transported to the size of the body only depends on a single coefficient that's shape dependent. So for a sphere, half of its body volume is being carried with it when it moves from point A to point B. A cylinder, it's 1. So the idea is that the drift volume depends on shape, not size. Remember, the mixing efficiency issue, the challenge we had is smaller things were expected to be less efficient. Here, all we care about is shape. We don't care about the size of the organism or the object moving. Now, what does happen when you put in viscosity? So this is that model with no viscosity. The real world does have viscosity, and we would quantify it with something called the Reynolds number. You don't even really need to know what that is, except to say that this is closer to the real world situation of, say, a centimeter-sized object migrating vertically at a centimeter per second. So in that situation, you can see that the drifting is even more significant. And maybe this matches your intuition. If I show you a different picture here, that green curve here is that Reynolds number of 100. As the Reynolds number gets smaller, the water gets more viscous. So think of taking a spoon through water versus a spoon through molasses. As I pull up that spoon through molasses, the more viscous fluid is going to be carried more significantly by that spoon as I raise it up. So this viscosity actually enhances this type of transport, where it was our enemy in the case of that other type of turbulent mixing that we talked about previously. So to show that this, again, was not just theoretical, Jana Narath, who's at a company here in Boston, actually, for her thesis, did some wonderful measurements in the laboratory. So this will come into focus in a second. The animal started down here, and it's very neat to see as it swims, it's carrying with it this quantity of the surrounding water. And ultimately, at the end of this video clip, for example, the length scale that matters here is how far this flow is transported, which is much bigger than the size of the organism itself. So the picture that then emerges is that you could have, for example, these vertical migrations. They occur everywhere in the ocean constantly. Somewhere right now is sunset, and you're having animals that are migrating from depth to the surface. And through this Darwinian mechanism, that could, for example, than single animals by themselves. That can then overturn where now the scale of these eddies here is much larger than the individual organisms. It actually scales with the horizontal area of the group that's migrating, which can be a kilometer wide, as opposed to, of course, the centimeter or millimeter sized organisms. And also scale migration distance, again, as Walter said, order kilometer. So this work gave us the sense that there could be biology at play in the ocean that's not the current one-way picture, that it's not just the animals along for the ride carried by the ocean currents, but that they themselves could be impacting the ocean itself. But it raises a lot of unsolved questions. In which context will this matter? We study the jellyfish sort of out of convenience, but there are other organisms that are more numerous and frankly located in climatically more important locations, and we'll come to that in a bit. Where in the ocean might this matter? In which seasons? What transport effects could matter? And then, of course, what could potential broader impacts be? This is, of course, not included in any of our models for how the ocean works today. We know that effects like overfishing, harmful algal blooms, acidification, eutrophication, all affect the biology in the ocean. And right now, we typically think of that as a one-way effect. We have a negative impact on the organisms. But what if the organisms themselves are impacting the surrounding water column? Now you have a much more complex coupled system that we really don't understand. So for the rest of the talk here, I just want to give you a sense of what some of the questions we've been pursuing are with respect to the context. And we wanted to switch to a different type of model system, again, one that is frankly more common in the ocean, important regions of the ocean, like the Southern Ocean. They exhibit large vertical migrations. This number, again, is still under debate because we don't have a lot of observations that deep, frankly. And so that, again, it emphasizes the need for better data. But the other reason we wanted to study them is because it allowed us to do things in a controlled, Ph.D. This summer. So we're using brine shrimp. These are the little sea monkeys you can buy at your pet store, and frankly, you could do some of these experiments there. Don't tell NSF that, because we spent more than what you can pay at a pet store for this. But for a lot of other reasons that I'll go into. They're easy to culture in the lab, these collective swimming behaviors. They have a phototactic response. So this you can do at home. Take a laser pointer if you have one and shine it down. As you'll see in a second, we're going to shine a laser pointer to the bottom of this container that's sort of yay big. And these little speckles here are those brine shrimp. And so you'll see as the light comes on, they all swarm to that burst, and so they all migrate together toward the top. And so this was an early demonstration that we could potentially generate migrations in the laboratory on demand, rather than having to be outside and wait only at sunset every day to do one-off experiments. So here's the schematic of the first setup that Monica put together. So we have a tank here. We have a blue laser that you're going to see in a moment turn on, and the animals love this blue light, so they're going to hug the sidewall here. Now, for these measurements, we don't want them too close to the wall. In the real ocean, you don't have these walls. So instead, they have a green laser that helps to center them, and then they don't respond very strongly to red. So we're going to measure the flow using a red laser. So this is just an animation of that. The animals are put in here. As Monica will tell you, the experiments are never this easy, but in the animation, you can make them do what you want. And then the idea here is that we make the blue laser rise, and the animals take chase. And so we can get them to do this dozens of times in a day instead of, again, the once-a-day measurement or once-a-day occurrence that happens in the real world. So here's just a photo of the lab experiment. I wanted to show with the lights on, because in the video you'll see in a moment, you'll see two beams, but that's just the refraction off of these two panes here. So here you're seeing that blue laser light rise. You'll start seeing some speckles in the green here. Those are the Artemia that are taking chase after the blue light. So they're migrating vertically. And then you can also see the red laser here. This is where we're doing a bunch of our measurements of the flow as this process is occurring, to be able to study what happens during the migration event. So the big picture questions that we have here are, do the animals actually create these flow features that are bigger than their own bodies? Even though these individual organisms are a few millimeters, could they possibly be creating flow structures that would matter in an ocean that's hundreds of meters, thousands of meters deep? So what you're seeing here is the flow of a single Artemia, very zoomed in here. And what we've done is to use a technique where the density gradient is changed by varying the salt concentration in the tank. It's very similar to on a very hot day, you're sitting at the stoplight or just in traffic in California, and you can see the shimmering effect of different temperatures of air, which have different densities and therefore refract light differentially. So this animal swims downward, it creates these vortices, kind of like what we saw in the jellyfish, and then because it had pushed heavier fluid upward, that heavier fluid then starts to sink back down. So one of the questions we have in the ocean is it possible that those density gradients that naturally exist would simply quell any animal-induced motion, that things would just settle back out to the way they began before you would get any larger scale flow effects? So we started doing these experiments, and what you're going to see in a moment is a zoom out. So the tank is actually over here to my far right, and we're looking to the side of it. And this was one of the first things that Isabel saw in her research, was that she would see these glimpses of these eddy structures. So the animals are chasing upwards. See those puffs that were generated. And those individual puffs were certainly bigger than the individual animals. But from this view, with this experimental technique, it was really difficult to get a sense of what was going on. So we switched techniques, or I should say she switched techniques, and developed a really clever experiment. So what we're going to do in this case is have the animals chase vertically after the light that they like, this blue light. The water's transparent, so the way we can see the water motion is we put these little tiny glass beads in the water column. So these dots here are just suspended particulate in the water. What you're going to see in a moment is the experiment begins. The animals are going to start rushing up toward the light as they would during a vertical migration. And I want you to, though, to try not to look at the animals. They're cute, but don't look at the animals. Look at the background flow that they're creating. So occasionally one will go through the laser sheet. It kicks a bit of the flow. After a while, more animals go, and that same bit of water gets kicked by the next animal. And so then you have this upward stampede of the animals creating this very strong downward flow that just gets stronger and stronger and stronger, 10 times faster than natural upwelling that oceanographers do care about in the ocean, 10 times stronger than what you would get in a natural wind-driven, say, upwelling event. So let's zoom out a bit more. So here you can see the box that we're looking at. We're going to look at the side. So on the left-hand side, you get this downward flow that we just saw. Again, that water, though, was lighter than the surrounding flow. So it wants to re-stratify, as we call it, go back to its original position. But in that re-stratification process, you start to get these eddying structures that are much bigger than the individual animals. Here's a little guide for how big the individuals are. You can see these being much larger. And then one more picture I'll show you. So in this experiment, what she does is to just take a layer of dye and has it sit at the top. So it's positively buoyant. If we do nothing, that dye just sits at the top of the tank. Now she starts her experiment and has them rise up. And the question is, how far can these animals pull that dye through this tank in this process? And so you can see here, sped up 10 times, but nonetheless, a very strong downward flow is generated by these animals by their upward motion. To get a sense of scale again, the individual organisms are casting these shadows in the laser sheet. So they are little tiny specks relative to this very huge motion that's being created during the vertical migration. So at the end, we can conclude that yes, the animals can create these flows larger than their bodies. And in fact, we find that as long as there are animals there to keep kicking the water further down, the flow continues to get propagated through our entire tank in these experiments that we published last year in Nature. Now in the lab, of course, we're limited to a finite size of tank. We can go 2 meters tall, and that's as big of a migration as we can fit. In the ocean, these migrations have been observed to be 10 to 100 meters deep. So if the same types of flows are occurring there, that has potentially very significant impacts on the transport of heat, of salinity, of various chemical properties, carbon, oxygen, nitrogen. We've done our own experiment showing how these migrations can alleviate hypoxia, so regions of the ocean that are low in oxygen concentration. And then think about also the community that is the ocean, the bacteria and archaea and viruses. We often don't think about the fact that they could be transported to new locations in the ocean with different environments and consequences for their own microbial communities by interacting with these vertical flows. So the last question I'll talk about today is just a question of what happens after that flow gets pushed down. Maybe it all sloshes back to where we started, and at the end of the day, our coffee isn't mixed at all. In fact, we've just shown some cute videos. So the way we tested this was to do a longer-term experiment. 120 minutes, which is roughly the amount of time that a little bit of the ocean would be kicked by a bunch of organisms as they go through that depth in the water column. And so basically we just have the animals swimming back and forth, and we wanted to measure how the density profile, the density as a function of depth changes. And so here you can see in the black curve, this is the shape of that density profile. Higher density at the bottom, lower density at the top of this tank here that's about 0.7 meters. After those 2 hours, the density profile is permanently changed, where the density is higher at the top and lower at the bottom than it was when it started. I won't go into too much detail here except to say these are histograms, the initial and final densities that we see in the water. This is the standard way that we would prove that we've actually irreversibly mixed the water. We've really physically changed the structure by this effect. And in fact, what you can show by mapping out how you go from the initial to final is that this is 1,000 times faster than just pure molecular diffusion. So this is a significant effect. Again, if we think about the wind and the tides, the wind occurs at the surface. The tidal effects can occur over surface topography at the bottom. These right now are the only source of mixing that actually is occurring directly mid-water, in the water column. And for that reason, I think it can also be significant. So what I would suggest is that the next stage in this work is trying to find direct evidence. We've sort of pushed the limits of what we can do in the lab. But again, if you talk to our colleagues at WHOI, they're not going to believe this until they can see it in the real ocean. And so there are a couple of bits of evidence that we can point to. Tarling and Thorpe, they're from the British Antarctic Survey, just last year, or 2 years ago, I should say, noted in their paper, I'm just highlighting one part of it here, they saw a pronounced downward vertical anomaly within swarms of krill in their case. So the challenge is many times people are going out to make these measurements. They don't want to study the krill. They're looking for properties of the water. This is a nuisance to them. But it's interesting, and it begs the question of whether we can use some automated techniques to revisit a lot of the data that's been collected over decades. And some of our colleagues are now doing this to try to find evidence of these flow features that were previously thought to just be noise in measurements of other processes. We're very interested in developing new in situ tools. So here, this is what Kakani has been doing lately, a technique she calls deep PIV. So taking that technique that she's developed, putting it on remotely operated vehicles down to as far as 300 and 400 meters to be able to do those quantitative measurements. In our lab, what we've been trying to do is to explore the possibility of separating ourselves from the need for shipborne measurements. So here at NBL over the next couple of weeks, Nicole Hsu, who's here in the audience, you can ask her lots of good questions about this research. What she's developed is technology that allows us to hijack is too strong of a word, nudge the swimming of jellyfish per different swimming frequencies. So what we can do is embed this device on an animal, and rather than it normally swimming as it does here on the left-hand side, so these are its natural swimming behaviors, we can ask it politely to swim at, say, half a hertz or 0.88 hertz and study right now the biomechanics of this process, but in the longer term, they could potentially provide a way for us to explore the ocean much more inexpensively than we do now. The shipboard measurements that we would do alternatively will cost you anywhere from $10,000 to $20,000 a day for ship time, whereas these devices she's developed are less than $1. So if we're able to develop these out as a source of distributed sensing in the ocean, it could potentially allow us to learn much more about the ocean much more quickly than we can now, to go from the 5 or 10% of the ocean we know something about to a percentage that allows us to answer questions like this on a much more reasonable time scale. And then lastly, I think it's important to think not just about fluid mechanics, that's of course my passion, but there are other signatures in terms of the biological and chemical signatures that could also indicate these effects, as I sort of hinted to earlier. And so I hope that those of you who are interested in the biogeochemistry might think about how your work might actually be the way that we resolve this. In terms of the fluid dynamics, I have to kind of be there and see the flow moving as it happens, whereas the changes in the chemical composition or the bacterial composition can many times last much longer. So with that, let me just acknowledge a lot of people who've contributed to this work. I want to especially highlight Sean and Jack, who have been extremely gracious in hosting me here for the past 15 years. They really are role models to me in what it means to be, first of all, brilliant scientists, but also very generous in their science. I think sometimes you get the idea that in order to do well in your field, you have to sort of step over the others in your field, to be stingy with your data, et cetera. And they've shown that that's certainly not true. And I think they're wonderful models for MBL and for science in general. I've been fortunate to have great students working on this and partners at Stanford. A bunch of graduate students who've contributed to this project and to others. Megan Leftwich and Brad Gemmel, who are here, brilliant scientists working on various aspects of biomechanics. I'm sure they'd be glad to chat about that too afterwards. And funding, because, well, science can cost money sometimes. So with that, I want to leave you with this quote from Walter. He unfortunately passed away earlier this year, but for me, his legacy lives in many different ways, not only in the science that he contributed over a career spanning more than 70 years, but also in nuggets of wisdom like this one, to really pursue those ideas that you most enjoy and to find that strange things can happen. And I'm certainly finding that in this work. So thank you for your time, and I'd be glad to chat more.
Thank you. That was really an amazing talk. My only frustration was twice you said that you would get to something later about how you get in the water.
Thank you, sir, I guess. At some point I was going to interject, so it's my secret shame, we're all friends here, so it'll stay here, that I can't swim a lick. I can't swim to save my life. In fact, the deepest I've gotten in the water here was over at Sandy Beach next to Stony there, a popular science article. They wanted to do a feature on the work we were doing, and so they wanted to do a photo shoot. And so they came out and they said, so do you have some ideas for some shots? And I said, yeah, sure. Why don't we, get Jack and Sean out there diving, and maybe I'll look over pensively at them. And he's like, that doesn't make any sense. Just get in the water. We'll take some shots. Just maybe float a little bit and pretend to do some science. And I said, oh, I can't swim. And he gave me this look like, kind of like the judgy look some of you are giving me right now. And so the best we did was I sat about a foot deep water. Jack and Sean had me hold a jellyfish, which meant I got stung everywhere. It was awful. But all that's to say, this work would not be possible without awesome collaborators who also know how to swim. Thank you.
Hi, John. Millions of people on the Earth, and all this traffic every day going back and forth. Do you think it affects the weather?
So certainly we know that there are indirect effects of transportation, for example, the carbon emissions associated with them. In terms of a more direct impact than that, I think it's tricky to separate, for example, the flow that a car makes from the ambient weather. So I think the indirect impact in terms of carbon emissions would be more significant for long-term climate as opposed to weather. Does that answer your question, or did you have something else in mind?
Well, I was just really wondering whether or not, in analogy with swimming fish, billions of people moving in cars ought to have the same kind of effect on the weather, if you think that's important.
Right. So the difference, I would say, it's true that there are large numbers, and here you have really even trillions of organisms. One of the key variables in this effect is the spacing between them. And so here you can have a billion organisms that are migrating together, all much closer together, though, than our automobiles typically are, just because the lateral spacing is typically quite large. So you might be bumper to bumper, as we are in California most of the time, but that's still not close enough to have the type of effect you can have when you have these groups very close together. So that's the main distinction in terms of the physics. The cars aren't close enough together to have that quite the same effect. Now, there are analogies. The Tour de France, of course, you'll see the cyclists close together, and they'll take advantage of the aerodynamics of their neighbors for effect, but that's at a much smaller scale.
Another question over here.
Yeah.
Thank you very much. This was not only informative, but it was really wonderful and entertaining.
Thank you.
My question is, does the upward migration affect the deep sea animals, sending chemistry down to them? And also, I wondered if the deep sea animals are creating mixing for the animals closer to the surface.
It's a very good question. So we have in recent, let's see if I actually have a slide here. So one of the things we've been interested in studying is those other effects of the animals besides the change in the density. So for example, we know that when these animals swim in a location that's hypoxic, here I'm showing you oxygen as a function of depth. Again, this is in a laboratory experiment, but we studied what happens when the animals swim through an initial oxygen layer that looks like this. So you have some higher level of oxygen, and then for various reasons in the ocean, microbial communities, et cetera, you can have a region of hypoxia. As these brine shrimps swim through that hypoxic region and they mix it, now you can see the oxygen levels at the top of this hypoxic region become much higher, in fact, much more habitable for a lot of organisms. So we do expect that there could be coupling when you're swimming through interfaces like from a hypoxic zone to a zone that's better oxygenated, where organisms above could have an effect on those below and vice versa. Now, to understand the extent to which this effect matters, we really need better data for how far the animals are going when they go down at night and where in the ocean that's happening. We have some sense of this from acoustic data, but again, it's like trying to understand the dynamics of this audience by talking to one or two people. It's just very difficult with ship-based point measurements to build up a meaningful picture. Yeah.
Toward the end of your talk, you showed bulk flow induced by organisms rising in a column. Do you have a sense of how well that how effective that will be when the migration isn't localized in X and Y? But he's basically got a whole plane of organisms rising.
I'm sorry. Yeah, I should have clarified. So what you were seeing was a two-dimensional slice. So our imaging is two-dimensional. The animals are migrating in a volume. So this is a large tank. They're migrating throughout the tank. We're just able to image through a slice of that full migration. So this is a three-dimensional vertical migration. We just don't yet have the experimental tools. We're trying to build them to map what that full 3D effect looks like yet. When we looked at the irreversible, and this is a good point, one of the referees wanted us to look at the irreversible mixing for exactly that reason, because we could only see what was happening in that slice. The measurements like these, for example, are volume averaged. So this is the effect throughout the entire three-dimensional tank in terms of how the animals are affecting the profile, not just in the plane that we image. Thank you.
Another question back here.
Do you think the animals are adapted to swim in those patterns specifically to mix up the ocean to benefit themselves and other animals in the ocean?
That's a wonderful question, and thank you. I'm glad to see a student question here. If there's others, you should take after him. So what we find is that, as this image suggests, there certainly is a benefit to be had in many cases. So the simplest example is these organisms that might feed on phytoplankton. So if they're able to bring up nitrogen, for example, or other missing nutrients to help support the growth of the things they feed on, you can see a virtuous cycle there. Now, at this point, it's speculative. What you really need to do to prove it is to measure those profiles while the animals are migrating, and at the same time to be able to demonstrate that it's due to the animals and not due to wind forcing or other natural upwelling events. So the possibility is there that occurs. To prove it is really difficult in the ocean. We just need to get more clever in how we attribute the mixing due to the animals from the mixing that's occurring from all the other processes. But great question. Thank you. Rick.
Yeah, 2 points. The images, the movies you showed suggest that it's not the Darwin entrainment mechanism, it's really the wake of their thrusting upward that's pushing fluid downward, so that they're not pulling the fluid along with them in their wake. Would you agree with that? I mean,
yes. So I glossed over this. The mechanism in terms of whether the net transport is up or down depends on the swimming mode. So in jellies, it's upward. In brine shrimp, it's downward. There are actually models that show that you can tune, depending on whether it's a puller or a pusher, as we call them, whether one happens versus the other. It really doesn't matter for mixing, though. It's as if you ask the question, am I mixing my coffee into the milk or my milk into the coffee? At the end of the day, it's mixed.
okay. The other point is you using salt stratification, I assume?
Yeah, that's right, in this case.
I would suggest if you thermally stratified it, you'd see even more mixing.
You're absolutely right. So one of the points raising here is this idea of partial mixing. So a salt will diffuse much more slowly than temperature does. And so if this was a thermally stratified system, as it often is in the ocean, then you'll end up getting parts of this flow that currently sort of sloshed back to where it originally came from and didn't mix. Those parts actually would mix even more effectively. And so I think because of that effect, these are conservative results in terms of the effect that you could see in the real ocean. Thank you.
I was wondering, I know in your model organism you use light to attract and make the movement.
Right.
What would happen if you use a predator that eats these organisms? Would that movement be a lot greater?
It's a great question, and in general, we've used the simplest mechanism we could think of in order to get the migrations to occur. In the real ocean, it's also worth pointing out, if a migration starts to be had, the moon, for example, is not penetrating down to a kilometer. So in many cases, we don't actually know what the behavioral trigger is. Maybe it is predators that are coming that's causing the trigger in the real ocean. In the laboratory experiments, that'd be a cool experiment to try, but it would be a more difficult thing, I think, to structure to have a predator. The other thing I would point out, in terms of how they plex, they might not necessarily go straight up to the surface. They could go up and to the side. So it would be difficult to structure, but I think you raise an important point that there must be other mechanisms besides light attraction at work, because there is no light, or very little light. No, there's very small amounts of light at that depth.
We have time for just one more, and then we can move the conversation to the next room.
I'm glad it's me. Thank you for your talk. Okay, so this is maybe a multi-part question, just sort of forming it out as I go. Let's assume that nanotechnology is advancing. Let's assume that propulsion, the behavior of propulsion has a wide application. First question, would you please describe the overall shape of this herd of microorganisms that you studied? Let's assume that it's possible to replicate such a herd artificially.
Right.
And that, as you talked about, sort of the density of the microorganisms is important in terms of to amplify their hydrodynamic effect.
Right.
So what is the shape of the herd? And then secondly, let's assume that it's possible to change the shape of the herd,
right,
and then put that on a surface vessel, like all these quote-unquote artificial microorganisms in a enclosed space in a free circuit of water. How might the change of the shape of the herd affect the flow that you have studied?
It's a great question. So to answer your first question, we don't really know in the ocean. So what you'll get typically is that I showed that map from Eric Kunz's paper where they had acoustic backscatter data. So you have a region of a dark band that was migrating. It was one of the earlier slides in the talk. So basically, you'll have a sense that there are organisms there, but it's very difficult to calibrate the acoustic backscatter to the individual animal separation that are occurring in that group. So you can say krill group, no krill group, but it's hard to say they're at 100 per cubic meter versus 200 per cubic meter within that group. There are people, Kelly Benoit Bird at Monterey Bay Aquarium Research Institute is trying to develop techniques to do that. So in the ocean, it's difficult to say. In a camera measurement, so they do have remotely operated vehicles where you can have a view of that. But it would be, if I asked everyone to stand up and run around, that'd be kind of fun. But then tried to quantify what's the spacing between people, it's very heterogeneous. And so what typically people will report is a range. What are the closest spacings you see? What are the largest spacings you see? And then in the experiments in the lab, we just take the average and try to reproduce that. So the number densities we use in the laboratory experiments are an average of what are seen in those other experiments. Now, what we're developing currently is a technique that would laser scan very rapidly, almost like getting like confocal microscopy or a CT scan, to reconstruct the instantaneous three-dimensional positions of all of the organisms. And then we can get at your question of where they're oriented one to another. To replicate that with microorganisms would be tricky for a few reasons. So the animals themselves, of course, they're propelling themselves on a trajectory up toward the light, but they're being influenced by the wakes of all of their neighbors. And so it's not that the organisms have an inherent control over that spacing, it's the emergent result of their own propulsion and their interaction with the neighbors. So you could probably approximate the average spacing, but to get an exact duplication, I think would be quite difficult. And at the end of the day, I think what we're looking for primarily is a lower bound. What's the minimum number density where you see this effect? Because then we can go into the ocean and say, okay, these organisms are too far apart. Like the earlier question about automobiles, they're too far apart, so we don't expect to see an effect there. These organisms over here, they're above a threshold where we would see an effect, and to start partitioning on that basis to begin with. Thank you.
Let's thank Dr. Dabiri one last time.
Thank you. Thank you. Thanks a lot. Thanks for having me. Appreciate it.